Brain implants are becoming more and more common, but how to make them safe and stable for many years remains a challenge. I've talked about this and much more with Vasiliki Giagka in this new episode of Bioelectronics Talk. Vasiliki is Associate Professor at TU Delft and Group Leader in Berlin at the Fraunhofer Institute IZM. I had a great time discussing with Vasso the present and future of neural interfaces. I hope you'll enjoy. Hello Vasso, how are you?
Hi Matteo, I'm happy I'm here today.
Thanks for being with us today.
Thank you for coming.
I am excited to talk to you about neural interfaces because your work is excellent but also very holistic. So I think we can touch on many subjects in a very comprehensive way. Tell me about your lines of research and what you've been up to lately.
Yeah, so in my team, we have two lines of research. The first one is translational. So it relates more to things that will find their way to the clinic. So we take into account several considerations related to the biocompatibility of the materials that we use, the ease of use, but also the longevity, which is a big part of our work, the longevity of the implantable devices that we make. And then, but also we also are looking into things that relate to how available the materials that we are using are eventually if these will be commercialized, which is something that is often overlooked. Not all materials are available for use in medical implants, even though we can use them in the lab. And then there is a second line of research where we focus on exploring new techniques to improve the spatiotemporal resolution. So that means how fast and how accurate we can pick up neural activity or activate regions of interest within the body. And we do that. So for that, we develop tools mostly for neuroscientists to use, where we integrate several modalities. So we call that multimodal neural interfaces. We integrate several modalities, particularly first electricity, light, and ultrasound. Each of these can give a unique dimension, and when we use them together, we can probe into the neural tissue with more accurately or more precisely. So we do that first as a tool for neuroscientists, and we learn also by studying or understanding the interaction of these modalities, with the goal that these eventually will also find their way to the clinic.
I will go back to a few of these points later, but I want to ask you how this recent explosion of startups and neurotech companies that went to the market and to clinical trials created a lot of excitement, also a little bit of hype. Did it boost the academic research or consumed all the oxygen from it?
So that's true. I have been in this domain since 2010. I started 2010 when I started my PhD in the UK at UCL. And there, one of my two advisors, Professor Nick Donaldson, was working for years in the domain of neural interfaces. So he was leading the implanted devices group at UCL, and he has a long history in this domain, even from his father. Mm-hmm. When I started, I think I was one of his last PhD students. So eventually Nick retired a few years after I graduated. So I wanted to say that when I started working in this domain, there were really not that many activities in neurotechnology. It was this niche, few researchers in the world. It was really not this field that we see today. And then when I was finishing, I think that was the year, so 2015, that was the first peak that popularized the field a little bit. So saw GlaxoSmithKline, so GSK, which is a pharmaceutical company based in the UK. We saw them getting into the domain of bioelectronics. So back then, the field, we called it electroceuticals, so this is apparently trademarked, so we're not supposed to use it anymore. So it became bioelectronic medicine. And it was back then that GlaxoSmithKline created this Bioelectronic Innovation Challenge. And they funded 10 teams in the world, and we were one of them. So this was what I was doing as a postdoc. So to address this challenge. This challenge was something that is so challenging that today we have not even solved it yet.
What was it?
So it was, so there were, if I remember correctly, there were 3 particular topics. So we had to develop systems that can record and stimulate neural tissue more or less in a closed manner, more or less real time. Or maybe it wasn't real. Maybe the requirement was not real time. This I'm not sure. But from peripheral nerves, from small peripheral nerves. So very challenging. So we had initially 6 months to debug our idea, the most risky parts of our idea, and then a second round to work on this. For the second round, I wasn't there. I was already at TU Delft. I had started my own research team at TU Delft. But for the first 6 months, I was there working on this challenge. So the fact that a major pharmaceutical company started this initiative and brought all these researchers together to address this challenge worldwide, so the challenge would give you 1 million euros, if I remember correctly, or dollars, if you solved it. The fact that they put all this together it basically created a lot of momentum. So that was, I think, the first peak that we saw. Mm-hmm. And it created a lot of momentum. It put all the noses in one direction, and it created synergies. And because suddenly there was money, there was also people from different parts of the world that they were being forced to work together to something like this. So that was one thing. And then beyond that, we saw after that more and more investments and more big companies getting into the domain. So of course, a pharmaceutical company, because of their sheer size of those companies and the amount of budget that they have to work with, is accelerating the field. But then we also saw this with others. So I think a couple of years after that, first of all, GlaxoSmithKline became Galvani, which was a synergy between so this group from GlaxoSmithKline and Alphabet, Google. Mm-hmm. And few years after that, we also saw other pharmaceuticals. For example, in Germany, we saw Merck getting into the domain of bioelectronics. And we can talk about that also later, if you want, when we talk about the neurotech landscape within the EU.
You work with Nick Donaldson, who, as you mentioned, was one of the early thinker about what then became neural engineering, maybe. Mm-hmm. For sure in Europe. Mm-hmm. How is he?
He's very inspiring, Nick, because he's a very humble, a very polite person, and he is really deeply interested in this. So the thing I always say and I really admire in Nick is that when I was in the lab, in the clean room, we had a small clean room for implanted devices, which we managed ourselves basically. And so, but he was next to me working on his own things at that stage of his career. So he had his own things in the lab, working hands-on on these things. So that's very inspiring. I wish I could do that, in fact.
Which one of the approaches of these big companies, and I mean really Neuralink, Synchron, Which approach do you think is more promising?
So now we see a variety of approaches. So we see approaches that focus on implanted devices, which is where I also come from. And there we see the effort to be able to record, stimulate from many channels in the body, but also distributed. Maybe we have not seen that in a commercial setting yet, in fact.
What do you mean by distributed?
Distributed, I mean not from one location in the body, right? So increasing the number of channels or the number of recording or stimulating channels in a neural interface can be for one specific neural population, but it's also interesting to be able to do that in several locations because several of these pathologies do not necessarily depend or being controlled by only one neuronal population that is in a specific location in the body. So, for example, I don't know, think about different areas in the brain that control different functions, language, for example, and so feedback maybe is manifested in a different area in the brain. It's also relevant for other parts of the nervous system. So getting stimulating peripheral nerves, the vagus nerve, and looking at activity on the brain. So you can have these distributed systems inside the body. The idea has been around for a while, but there are challenges in making it work. So I think commercially, if I'm not mistaken, we have not seen that. Maybe Mint Neuro is looking into this, but they are new. So I think there's nothing advanced in this area.
Mint Neuro is in the UK?
Is in the UK, yes.
These that we mentioned are mostly invasive, but there's also a lot going on into the non-invasive realm.
Yeah, exactly. So so we have the heavily invasive, and then we have approaches that try to minimize that. So from less invasive to, minimally invasive or non-invasive. So of course, it goes without saying that, at least so far, the non-invasive techniques are suffering in terms of spatial resolution. But there are approaches that are working to increase that, and maybe with the emergence of AI, maybe that also accelerates things, because maybe we will be able to. I mean, there are also a lot of issues on the hardware side before we're able to harvest AI to process the data. But on the hardware side, there are also some bottlenecks there, because the amount of signals that we get, the signal-to-noise ratio is simply not strong enough to be able to. Let's put it this way: we can record better signals when we are close to the target.
When it comes to non-invasive and invasive, I often think that, in principle, we could do, for example, a pacemaker that is non-invasive, but I think that patients would still go for the invasive option, would still want a surgery for sure, but something that they cannot forget, something that is there and it's barely visible. So there's also an element of patient adoption there, and sometimes the invasiveness could be more favorable than not.
So that is another issue. Patients don't want to think about a device that they have to. It's like when you're taking drugs, you don't want to think about taking your pill every day. Ideally you have something that's in your body. It would work in the background without you having to do anything about this. Maybe adjust your dose every once in a while if it's needed, but that's it. And so that is indeed in favor of invasiveness. So there is where we are talking about minimally invasive strategies. So of course, something that is very invasive, brain surgery, deep brain regions, that's less appealing for patients. But something that's minimally invasive that they can have there and forget about it. I think that is a good solution.
You mentioned Mint Neuro, which is a UK-based company. And it's when I think of neural technologies, I always think of American stuff and Australian stuff. But it's true that there's good stuff going on in Europe. What do you think about that?
Absolutely, this is true. It was not like this in the past. So we've seen an increase in number of neurotech companies that are coming out from the EU. And I'm very happy about this because, also as an academic, we educate people. And these people, they want to be able to work on what they are educated on. So it is a pity to then have to tell them, okay, now you need to leave, emigrate, and go somewhere else. So, and I think there's a lot of potential in the European ecosystem. It has, so we've seen recently a lot of things in Germany, of course, Cortec. We have a Neuroloop from Freiburg. We have Merck, which is working with two other neurotechnology providers, and as a big pharmaceutical is pushing this forward. We have InBrain from Barcelona, and Inervia, the smaller sister company. We have Mint Neuro in the UK. We have Coherence Neuro. I don't know if you're familiar. They are also a startup in the UK. In the UK, Cambridge, and they're targeting cancer, if I'm not mistaken. Neurotechnologies for cancer.
NeuroPixel is from Belgium.
Yeah, that's it's true. Yes, NeuroPixels are from Belgium. It's part of IMEC.
And lots is also going on in Switzerland. Spinal cord.
Exactly. Onward.
Yes.
And Neurosoft Bioelectronics.
Yes.
Yeah. Yes. So a lot of things. It's true. And more will come, I think.
In Europe, often the problem, I feel, is not technological, but it happens later when it's time to go to the market or to the patient.
Yeah, that is absolutely true. So there's a lot of potential. There's a lot of good things are coming out. And I think the problem that these companies are facing is that at the moment that they need to go to the market and think about reimbursement strategies, in Europe, the market is more segmented. So we have different countries. Each of these has a different system. Health insurances you will have to negotiate with each country separately. And so, and unless this becomes a unified thing, I don't know how, it is always easier to have as an entry point the US because it is a big market in terms of numbers, but with one system. Yeah. So you don't have to go and talk to so many different parties. And of course, also the regulatory requirements in Europe are at the moment more stringent compared to the US. So these two things are challenges that European companies need to face at this moment.
I'm asking you about the European scene because you have a very European profile. Yeah. You're European, you moved across Europe, and now you have a double position in two European countries. How do you split your life and organize your life between this double position in the Netherlands and in Germany? And how do you organize your two research groups?
Yeah, I'm Greek originally, so I studied in Greece. I'm an electronic engineer by training, and then I moved to the UK, London, for my PhD. Stayed there for a short postdoc, and then moved to the Netherlands, to TU Delft. So Delft University of Technology, and there I started as a tenure track assistant professor. And then 3 years in, at some point, I received a personal fellowship from the Fraunhofer Society to start a new team at Fraunhofer IZM in Berlin. And I combined the two since 2018 until now, and this will continue for the foreseeable future. because it works very well in some aspects, I can explain. And so, indeed, so my team is one. It is one team. I don't consider it two teams, it's one team. We always meet together. I call my team SOMA, which stands for Soft Multimodal Neurointerfaces. And this name it is a Greek word, soma. It means body. And it is in fact the body of the neuron. And the body of a neuron is also where impulses are initiated, and from that point travel across along the neuron and to adjacent neurons eventually. So the fundamental things regarding neuromodulation, basically.
Europe is this relatively small place, but very diverse.
Yeah.
And, different places do things differently, socially, but also professionally. So it's different to be a professor or a supervisor or a student, or how to write a paper or grant. Is there something that you adopted in one of these countries that you still use for you or for your group when it's time to teach or be a supervisor, something that worked particularly well for you?
It's true. I've seen different academic systems, and at Fraunhofer is also a research institute, so it has also a slightly different focus. It works a lot with industry and applied research. So I've seen different things. I've seen the purely academic thing part. I've seen also the more applied part. And yeah, I can say that from all these, there are things that I keep with me. So, for example, in Greece, it has been a taboo for universities to work closely with industry traditionally. Universities are considered places where a lot of thinking is happening, and this should be protected, and this should be left uninfluenced, let's say. And traditionally has been the mentality that it's not good if companies are intervening with this process. And then I saw how this is very different, for example, in the Netherlands, where a lot of the work that we do is even directly funded from industry. I see how this accelerates innovation. Then another thing that I've seen moving across or experiencing different systems is the pros and cons of being at a technical university, at a technical-only university. So Delft University of Technology is a technical university. And before that, I was always at UCL, but also in Greece, universities that are not purely technical. And that's that was a realization that occurred to me only when after a couple of years in Delft, that it pushes innovation, of course. It also, because everyone is an engineer, so we speak more or less the same language, we understand each other, and the needs of research teams are more broadly and better understood, because we need infrastructure, we need specific type of funding and this kind of thing. So that makes it easier. On the other hand, there's always a need for the other voice. There's always a need for people that don't speak that language, because they bring in a different perspective to the discussion. And I think technology without this discussion is limited, or maybe eventually going in the wrong direction.
You mentioned at the beginning that you try to realize solutions for neuroscientists. So you have to talk to neuroscientists.
Absolutely. But it's not only neuroscientists. Neuroscientists are, so absolutely. And this is where we also see difference in the language that we speak. For example, engineers. For engineers, things are measurable, things are more specific. Even at a different scale, so micrometers, nanometers, we can monitor them, we can measure them, we can be accurate at that level. In the body, nothing is like this. In the body, things are approximate, and few millimeters here and there, variability will always be there. How big my vagus nerve is compared to how big yours is going to be, and also along its length, that's going to vary. And so when we talk about these things with neuroscientists, there is always some calibration that needs to happen. So we need to know how accurate they are, and they also need to, it's always
the important questions are different.
Exactly. But also understanding what it means when I ask how big is this, so that I know how to design my device, and I get an approximate number. Not to blame. In fact, I don't think so. It is just a different point of view, which I think is very, very valuable. But so neuroscience is one, but then it's everything else around society, right? So ethics. It's another important aspect in neurotechnology development. So we think about how to make things, but then how do we use them? These pose a lot of big ethical questions that I don't think engineers are the right people to think or discuss about these.
I've heard you saying every neurotechnologist should be trained into neuroethics.
I think so. At least they should know where they stand on this and what are the implications. I think technology development is not something that you can block or decelerate. You will continue accelerating. But you should know where you stand, and you should be able to appreciate where things are going in this domain.
When you talk about the electronics you develop, you put a lot of emphasis on the word active. You call them active neural electronics. What's the idea there? What does that mean?
When we talk about neural interfaces or neuroelectronics, which is a different name for it, often I wanted to make a distinction between having the electronics in close to the neural interface and not. And by active, in my case, I mean integrating high-performance electronics. That means CMOS technology. So the kind of microchips that you have also on your phone, in your phone. But it can also take the form of some electronics that are not so high performance, but are locally needed close to it to be integrated within your neural interface to perform some basic tasks, for example, pre-filtering or multiplexing, to be able to pre-process your signals so that they can later on be processed by more high-performance electronics that are a little bit further away. So these are components that so when I talk about active neuroelectronics, sometimes it is confusing for people because it brings a little bit of two different disciplines closer. One is CMOS technology and microelectronics, and the other one is soft polymers that interface with tissue. So that means fabrication of these soft neural interfaces. And these are normally people with different trainings, material science versus circuit design or electrical engineering background. And these don't, it used to be that they were a little bit distant traditionally. Of course, we've seen that this domain, this field is multidisciplinary, and in it being multidisciplinary, a lot of different disciplines have to talk together. You probably previously mentioned neuroscientists, which is even further away from these two. So people are learning to talk each other's language. But with active neuroelectronics, now I consider integrated microchips within the a soft polymer, which is used for the neural interaction.
Something more traditional, say a DBS probe using Parkinson or a pacemaker. Would you consider those passive?
No, these are also active. These are also classified under these are considered active medical devices when they are regulated. You have the electronics there. Okay, maybe this is a good distinction. So you always have the electronics in the body. In my field of research, we try to bring those closer because we're trying to miniaturize everything. But these are also active neural interfaces. The differentiating factor is that here in a pacemaker, for example, the classical construction of a pacemaker, you have the electronics that are housed in a hermetic environment, which is implanted somewhere, usually subcutaneously close to the chest. And then from that, you have connections, the cables that connect to that and go to the electrodes. And the electrodes are the contact point with the tissue. So where exactly we want to stimulate, this is where the electrode is. And this can be a long distance. So for the next generation of these active neuroelectronics, we are looking into ways of miniaturizing everything. So making the electronics much smaller, getting rid of the battery. So we're talking about battery-less devices, and concentrating everything into something small that is now minimally invasive. So going back to the overall picture, we are looking into minimally invasive techniques. So something that is millimeter sized, that has all the intelligence. And for that reason, we're also looking at alternative ways to package. So to package those neural interfaces so that it can also last decades in the body.
Very desirable feature for virtually any biomedical device is the ability to have a closed loop feedback. Is it fair to say that you need active electronics to create this coordinated smart loop?
Absolutely. You cannot do this with anything less than that. You need the high performance CMOS technology, and you need a lot of also strategies to do data manipulation. So absolutely, you need active electronics to be able to do that.
We mentioned before that recently many startups and big companies came to the field. And depending on the application, many of these companies need to produce implants with many channels.
Yeah.
Now, before this explosion of companies, you gave a talk whose title was, and I will quote, "Towards a flexible brain implant with 10,000 independent channels." This was in 2019.
Yes. We are not there yet.
What was the idea there, and where are we at?
Yeah, so there are applications you need this number of channels. For example, in the eye, to reach the resolution, to be able to see with enough resolution, you may need that kind of number. I mean, there are more. Another example that I can think of that will need a lot of channels is probably motor control. So you have a lot of degrees of freedom in controlling motion with your hand. And if you can probe so specifically into the nervous system, you would ideally want to use that capability to improve to improve movement. And for artificial hands or robotic hands, or your own, depending on the type of application we're talking about. And there are more. We are not there yet, and there are a lot of challenges in order to get there. There are challenges across domains. This is an exciting field for an engineer because there are challenges we can actually tackle. But there are challenges in the neural interface itself. So the soft implant or that interfaces with the body. How do we make 10,000 electrodes? how do we make them so that they last long enough? How do we make them so that they are small enough? They are implanted in a minimally invasive way. And in fact, the smaller we make the electrodes, we run into an impedance problem. How do we make them so that the impedance is also very low? And then from that, we need to, if we think about neurorecording or neuromodulation, in both scenarios, we need the electronics beyond that. So how do we pick up this neural signal at an intensity that is enough for us to be able to post-process it? Or how do we transfer this charge into the tissue to be able to activate with so small electrodes and so tiny electronics? How do we do that at a very low energy? So low power techniques. How do we do that at a very small, with an electrons that have only a very small footprint? And then let's assume we have the electrons, we have the electrons. How do we bring the two together? How do we connect 10,000 channels together so that we can independently or drive those or groups of those? Because I think independently, a single one we're not there. And then let's assume we've done that and we have the electrons that can do that processing in the body. How do we bring all the information that we get from 10,000 electrodes out of the body and make sense of it? If this is data that is being recorded continuously, we are talking about terabytes of data so how do we do that? So all these challenges span different disciplines, different fields, and there is a lot of people that are dealing with each of these challenges separately, so that we eventually, in some years from now, we can push it forward.
What kind of clinical insights we can get if we start, if you go beyond 1,000 channel and we go to 10,000? Do you think we get so much more information, or it's a bit of exaggeration?
We don't know that yet. That is another open question. I think there is somewhere a sweet spot. We probably don't need to do. I'm not going to talk about the 10,000 because 10,000 channels could be 10,000 channels that all record from single neurons, or 10,000 channels that record from the complete brain. Right? So these are two different scales. I think so I would like to reverse it and talk about resolution in the body. So single neuron resolution is probably not needed in the body, at least for therapeutic purposes. I don't think it's needed because there is not a single neuron that is not replaceable in the body. So that's probably an overkill. That's probably too much over-engineering. However, single neuron resolution could be interesting to decipher, to understand what is happening. And that's another challenge. We have not mapped our nervous system. We don't know exactly what is happening in our nervous system. Where are things connected? How are things being controlled? How do we think? These are questions that have not been answered. And to be able to answer those, probing at the nervous system with that high resolution could give you a valuable tool. Now, then you run into a problem that, yes, okay, assuming I can do that, what do I do with this data? Do this data give me something useful? And I think that's another issue that has to now be addressed, probably now with the help of AI. But until we have this data, we will not know how useful this data is. But for therapeutic purposes, I don't think that single neuron resolution is useful. Now, the 10,000 channels can still be useful though, but at a different scale.
Again, in this environment of new companies on the scene, I think among the excitement, one dark aspect is this. It's the fact that many of these maybe found a market that was too small and went bankrupt, which is maybe normal for startups. But some of these went bankrupt after they had implanted patients. leaving these patients with the foreign electronics in the body without support, technical or clinical or psychological. This is clinically and ethically terrible. And to make things worse, the fact that these neural electronics can break easily, leaving these people with a broken piece of electronics in the body. And you spent quite some time working on the reliability of these neural interfaces, trying to understand why they break and what can we do to improve this problem. And tell me what your research suggests.
There are two things I think we need to do here, and I will go into my research. But before I go there, I wanted to say that one is explantability. Mm-hmm. So whatever we develop, we really have to make sure that there are ways to remove it out of the body, to remove it from the body when it serves no purpose anymore. So that has to be embedded in the design of a neural interface. It cannot be something that we neglect just because of the reasons that you mentioned. But also maybe because it stops working. And so you don't want to have a piece of electronic in your body when it does not work anymore, because there are risks associated with it. But okay, how can we minimize these risks? This is part of my research, a big part of my research, in fact, in the longevity of neural interfaces. And so for that, we are looking into ways to package electronics so that they can remain in the body for decades. And this is not a challenge. This is a challenge that has been addressed in commercial devices by engineering packages that are themselves engineered to last decades in the body. And this is a solution, for example, that is used in pacemakers. This has served us quite well. And it will continue serving us for the foreseeable future. I don't see a need to change that. Because pacemakers fulfill a function that is really life critical. It's life critical, and it is then the delivery of the therapy is time critical also. So you need electronics to be able to respond at the specific time that you need them to in order to deliver the therapy, because the life of the patient depends on the timely delivery of the therapy. In that case, you need packages and you need batteries that can support and have this energy for your implant available exactly at the moment that you need it. Now, with the emergence of bioelectronic medicine like a decade ago, we have seen therapies, for example, bioelectronic medicine therapies that that aim to treat autoimmune conditions, for example, diabetes or Crohn's disease. And there are also other examples. The delivery of the therapy is not time critical. So we now move from a paradigm where availability of energy needs to be there at any given moment to a paradigm where we can deliver therapy a few times a day at low energy levels. And the timing that we do that is not critical. It's like a pill, you have to take it every day, but you don't have to take it at exactly that time, right? So now with this, we can move to battery-less implants. So remove the battery and deliver the therapy when we have energy available from external sources. So this gives us a lot of room to reduce the size of the implant. And now we can move away from that paradigm of packaging into a miniature, which takes up a lot of space, to a miniaturized version. And what is the most miniature version? Conformal coatings. So we are looking into conformal coatings with polymers and thin film organic and inorganic materials to use as a protection for miniature electronics now that we make minimally invasive, to improve the longevity, reach eventually with a goal of reaching decade lifetime for conformally coated neural interfaces.
this line of research is mostly material science, or it goes also in the circuit design?
Okay, very, very, very relevant question. Thank you for asking this, because that is the new insight that we discovered working on this topic. So in the past, for the conventional way, where we have a package engineered to last a decade on its own, and all the electronics are inside, the packaging is designed independent of how the electronics are designed inside. So these two fields don't meet. When me as a circuit designer, let's say I'm designing my electronics, I don't need to care about how this is going to be packaged, and the other way around. However, when we are talking now, we want to push the state of the art towards miniaturization, minimal invasiveness with conformal coatings. Now we cannot ignore that anymore. So now we found that conformally coated electronics or implants will have a lifetime that is defined by both the lifetime of the conformal coating, or how these conformal coatings interact with the components underneath, but also the way that these components are activated in the body, so the electric field that they are going to see. So that means that the circuit design of the complete implant will now have to take into account the fact that it's going to be conformally coated. So we've seen that, for example, one specific example, a DC electric field will cause faster failure in conformally coated implants compared to AC electric field. This is also intuitive, because if you have a DC electric field, you always push ions in one direction. So you are accelerating. With an AC, you have time to reverse from time to time. And so you lead to longer lifetimes. We've also seen that failure mechanisms are going to be different in the two scenarios. But what I wanted to say is that now, with the understanding that the electric field that the implant is going to see in the body is going to be different, it means that we need to design our electronics to be conformally coated, conformally packaged. And now by doing those two together, we can really push the longevity of those implants.
I find this type of research really commendable because it addresses a clinical need, an engineering need, and as a material scientist, I like what's going on there in terms of electrochemistry and diffusion and stuff. But particularly commendable because this is a lot of work that has to be done in very specific conditions, in vitro or in vivo, for a relative little amount of results. So it makes it maybe not much of a sexy academic topic. So not many people are pursuing that.
Absolutely. So maybe I would correct the little amount of results. The results are a lot. It's just that the probability of failure is high. But still, you understand things. But it's also that the timelines are long. So how do you test if you want to prove that something lasts for a decade or two? So you don't have that decade or two. So it is a complex topic, that is true. And it's true that it is not a very sexy topic in academic environments. For me, that was also the reason why I wanted to have the Fraunhofer affiliation. And the reason is that as a research institute, in a research institute, I have the possibility to do this over longer time frames, study this longer. I can have. So it doesn't fit very well with academic research because if we usually in academia we have PhD projects that last 4 years, and basically the results have to be there by year 3, and then people have to write their thesis and move on, right? And then often what happens is that a lot of the know-how also lives with the PhD student because of the uniqueness of the thing. And so, in a research institute, this is a little bit different because you don't have to stop at 3 years, and you can have this continuity. So more continuous setups, processes, protocols. So this part of research, whereas we do a lot of things in Delft to be able to understand more fundamental mechanisms, but we primarily. We can do long-term testing in Berlin, so that's how I organize this. And of course, new insights lead always to new questions, so we close the loop like this.
When I was a PhD, I might have been guilty of trying to pursue the exotic research and finding something cool rather than taking care of important pragmatic aspects such as stability and reproducibility, which is probably wrong, but also understandable if you're a PhD.
No, absolutely.
How would you convince a very enthusiastic PhD to take care of these other aspects? Less attractive maybe on paper, but still very important.
And just to say, I think it used to be not attractive. Mm-hmm. I think this trend is changing a little bit just because people now start understanding how important and difficult it is. So there is a lot of fundamental understanding in this. It's not that trivial. It's just that people in the past probably neglected it because, when we have to solve more basic problems, longevity is not the issue that everyone is looking at. But now, some of these other things have been solved, and everyone is looking at, okay, but now how do I make it last longer than a month or so? So I think it used to be not attractive, but this is a changing trend, in my opinion. And I mean, maybe in the past it was more complicated. Also, because I also understand if a PhD student starts, they want to work on something that can give them a breakthrough because it's their career eventually that is at stake with this, unfortunately, because of the way that we assess right now. But this is a different topic. But the way that we assess people and performances in academia, this is how it goes. But right now this has changed. So I don't think it's not that sexy anymore.
At the beginning, you mentioned you are also looking at multimodal interfaces, and you mentioned electricity, ultrasound, and optical. How do you combine them?
That's something that is more and more growing as an activity in my team. We started a few years ago, and we at the moment are touching up all these 3 modalities. So what we are doing is that we are looking into ways to combine electricity, light, and ultrasound, not all of them together, usually 2 at a time, to improve the spatiotemporal resolution in neural interaction. This is both for neuro monitoring, but also for neuromodulation. So I can give you a couple of examples. In the domain of monitoring of brain activity, it is possible to monitor activity from big areas in the brain or even whole brain for small species using functional ultrasound. So in functional ultrasound, we don't actually monitor. This is an analogous of functional MRI, but using ultrasound. So we now don't monitor specifically directly neural activity, but we can monitor change in the blood flow, blood flow dynamics, And we can correlate that to neural activity. So when there is neural activation in an area, that activity becomes higher. So indirectly monitor neural activity through that. And that can be done with functional ultrasound, which is different from normal ultrasound monitoring because it uses very different technology in terms of how the ultrasound is being directed in the body. But anyway, to cut the long story short, it uses ultra-fast ultrasound to do fast monitoring of areas in the brain and monitor blood flow. Now, to correlate that directly to neural activity, we want to be able to have neural interfaces that can pick up that neural activity so that we can do the correlation. So monitoring with functional ultrasound and monitoring electrically. And these neural interfaces now have to be acoustically transparent. So we have been working on understanding and simulating and putting down the requirements for neural interfaces to be made acoustically transparent so that we can combine both ultrasound and electricity in neuromonitoring. So this is one example. And then another example relates a little bit to the work that I do with graphene, together with a colleague of mine, Sten Vollebrecht from Delft University of Technology. We have been collaborating many years on this topic. And there what we aim to develop is optically transparent neural interfaces based on graphene. Graphene is an 2D material, and we use it that can create optically transparent conductive tracks and electrodes. And we use that to create a neural interface that is now optically transparent. Why is this useful? This can be integrated in vitro setups because it enables microscopy at the same time as electrical monitoring. It enables calcium imaging, so visualizing active cells when they are activated during electrical stimulation that is applied through the graphene electrodes, but also the other way around. So optogenetic stimulation through the optically transparent electrodes, where now the evoked activity, which is electrical in nature, is being picked up by the electrodes. So there are all these interplays with these different modalities. This is an exciting field for us and for my team. We see a lot of potential in this domain. So there are more that we are developing in this domain and more to come. We are also thinking into integrating thermal modalities in there.
What for? Cooling down or warm up?
Oh, there are several things. For example, temperature is also another factor in the body, and changing the temperature may change the way that neurons are. Well, will change the way that neurons are activating. If you look at the Hodgkin-Huxley model, temperature is a big part of it. So perhaps you can locally change the temperature and make activation more easier. So there are all these interplays that we are bringing into this multimodal regime. That is also very exciting field too.
We close our episodes by asking our guests to mention a book or a movie that, even if it's not related to their research, you would anyway advise everyone to read or watch.
Actually related to my research, and it is a movie which is called Cyborgs Among Us. Mm-hmm. Have you watched it?
No. I will.
It's very interesting. It is a documentary from 2017, a Spanish documentary. I mean, it's in English, but a Spanish production. I think. No, so let me read the name of the director because I don't want to have it wrong. It's Rafael Duran. And the movie is very... So we watched it a few years ago in one of the movie sessions that we did with the team in Delft. I think it was before COVID. And it sparked a lot of interesting discussion. So it is a very interesting movie because it goes from, so it covers aspects such as biohacking. So it goes from right now, the reason why, particularly for me, it is the case, we develop these neurotechnologies is to give people the capabilities to restore functionality that they had before and they've lost because of injury or because of condition. And this is a big, I think, discussion in this field. Where do you draw this line? So for me, this is the line that I would not like to cross, but there is also a lot of people that are looking into enhancement. And so enhancing your capabilities, what your body can do naturally, being able to now do more of it. And so this documentary covers efforts that are done in this domain, for example, in simple biohacking, that is very DIY, that people do in their basements with tattoos and these kinds of things that are trying to sense more things than they normally could. But also moving into actually discussing what is happening in the medical technology or neurotechnology domain and where this is going. So maybe right now is 2025, since 2017, this might be a little bit outdated, but it's still very relevant. And then it also, for me, which was the most memorable thing from this movie, moves towards discussing the ethical and societal components that relate to these developments. And so how these are going to affect societies when people have the capability to do more, who has access to this technology. This is now wealthier people that will have access to that. So that separation now becomes even more prominent, right? So it's not only wealth, but it's physical ability that you have more if we cross that line. And this is also closing the loop to what I said before about ethics. That is also very...
Cyborg among us.
Cyborgs among us.
We'll watch it.
Yes.
Thanks a lot, Vasso.
Thank you, Matteo.
It was a pleasure.
It was a pleasure.